So thank you so much for inviting me to this conference, and I would like to show a little bit a point of view a little bit different regarding to the applying the microbiome sequencing for in clinical, preclinical animal models for brain damage. That is basically one of the main research that we have in the lab, and then I will show you some of them. So I'm a neuroscientist, and as a neuroscientist, so we always study the brain as something isolated from the body, and that is not true. So a few years ago, we started to explore the periphery and the connections they have, the brain with the different organs, and we explored the liver, the spleen, and the lungs, etc. And lately, in the past kind of 7 years, we start to focus on the brain-gut and brain-gut microbiome. And as you know, for neurological disease, the inflammation and the immune system is quite important, and one of the major systems that they can regulate the immune system is the gut. Around 70% of the immunity, 70–80% of the immune cells, they have this connection with the gut. So this mechanism, this communication is quite important to understand some of the disease, some of the causes of the disease. You know that not always a low percentage of the cancers, etc., they have a genetic component, so there are environment components as the gut microbiome dysbiosis can have an important impact. We know that, for example, the liver infection, the cancer, liver cancer or heart disease, asthma, infection, obesity, diabetes, all these diseases, they have a connection, a link with the gut microbiome dysbiosis. And as well, the brain. Of course, the Crohn's disease and the Wall disease, they have a direct connection. But the brain as well, they were identifying mechanisms that regulate the microbiome, and they are involved in Parkinson's disease, in Alzheimer's disease, stroke, or even autism. So why this thing happen is that because for example, I will focus this talk on one of the animal models that we have in our lab, that there are traumatic brain injury and concussions. So when we have inflammation in the brain, when we need, when there are axonal damage or brain edema, there are immune system inflammation. So there are alterations in the neuronal plasticity, etc. So that this interaction as well has an direct impact on the gut. And that we know because we identify imbalance in the digestive system, vomiting, nauseas, etc. Decreased intestinal peristalsis, and all that, there are direct impact of the trauma or brain injury in the gut, and directly induce a gut microbiome dysbiosis. And these mechanisms that are regulated with different pathways, and we identify several of them. The immune neuroendocrine signaling that can communicate the brain to the gut, this bidirectional communication as well. The vagal nerve activation, microbial factors like products that are produced in the gut that are released into the circulation that can make this bidirectional communication constantly between the brain and the gut. And what happens in the epithelia, in the intestinal epithelia, so we have the immune cells, the T cells, B cells, and that interact with the epithelial cells, and we have microbiome bacteria that they are producing the short-chain acid fatties and all the neurotransmitters and all these compounds, metabolites, they are released to the circulation and can affect to the brain-gut communication, right? So this is the overall the mechanism that I can regulate this process, and so now we need to manipulate these mechanisms to try to protect the brain in some way. So in my lab we are studying different diseases that are involved in the microbiome, brain-gut microbiome connection. One of them is the traumatic brain injury. We did as well a clinical study with concussions in the football players, at Rice University, by the way, and we identify change in the gut microbiome after a single concoction. We are interested to study the long COVID and the neuroinflammatory impact of the SARS-CoV-2 a long term. So the microbiome plays an important role as well. And then we have Alzheimer's disease. We are using patient samples and then animal models for Alzheimer's to try to discover some kind of these mechanisms that are regulating. And for this reason, you see the mice that I have here in the back. So we have mice that we injure the mice. We have Alzheimer's mice that we injure the Alzheimer's mice. And we have different well-established animal protocols in the lab to study this disease. So our original hypothesis that we could present years ago, and the last time was a neurotrauma meeting, was we thought that repairing the microbiome, the gut dysbiosis caused by traumatic brain injury, would be enough to reduce the acceleration of the Alzheimer's pathology and other disease. So that is the premise. But first, we need to see which is the impact of the TBI, traumatic brain injury, in the gut. So you are familiar with the preclinical animal models, so it's perfect. And now I'm going to explain briefly what we did. We analyzed the gut microbiome in the feces of the animals before injury, and then after injury, we use to induce the injury in the animals, we use the controlled cortical impact injury that is consisting of a pneumatic impactor that impacts in the brain and then can cause a lesion, you can modulate the severity of the injury. And then we collect the feces at different time points. For the studies that I'm going to present here, we analyze the cystic X RNA gene sequence analysis using the Illumina, and then we did the microbiome analysis. For the clinical studies and other analysis, we use the nanopore sequencing and other bioinformatic approach, but for these ones that I will present, we use only the cystic X. And the first paper that we published was in 2018, and what we found here is that after a single TBI, a single traumatic brain injury, there are a depletion, there are a decrease, significant decrease, all the Lactobacillus family. And then at species level as well, we identified 3 or 4 that they were reduced significantly after TBI. That was just 24 hours after TBI, and then we look at the overtime and 14 days and 4 weeks after injury as well. The Lactobacillus was the most predominant species that were decreased systematically always after inflammation, after trauma. So we know that in neuroscience and neurological studies, we know that the Lactobacillus strains that are associated with attenuation of anxiety and inflammation in the brain, and then as well intestinal permeability. So it makes sense that this family, these strains, they were reduced after trauma, because the trauma per se can increase inflammation and intestinal permeability and cognitive decline. So what we know, that is great. Some of the reviewers and papers and grants, they always asking the same question: what happened in humans? What happened in humans? Because that were preclinical animal models with in mice. So we did several sharing the literature, and we found that the Lactobacillus that are associated multiple trauma patients. There are a decrease of this family as well. We found that the Lactobacillus family, they were neuromodulators and the levels of neurotransmitters. So there are a lot of association in clinical studies showing that Lactobacilli probiotics as well improve the cognitive function, et cetera, et cetera. We didn't did any clinical study with probiotics, but we want to do it first in the animals, and that is the reason that we ran the next experimental design. So what we did here, we have the same animal model that you can see here is the TBI, and then we administrate pan-probiotic cocktail of several lactobacillus, several species that we identified that were interesting for recovery and neuroprotection. Plantarum, Reuteri, Helveticus, all these lactobacillus, they were identified in clinical studies that they were beneficial, like for reducing inflammation and cognitive improvement. And then we did the TBI model, and we we analyzed the microbiome at different time points, and then we performed behavioral testing. I don't know if you are familiar with behavioral testing in animals, but there are several approaches that we can do. And then we analyzed the response acutely and as well chronic time point. So what we found so far is that the PAM probiotics, all these lactobacillus, they make an impact on the diversity, of course, that we expect. Especially in baseline, there were levels, similar levels, and then in 13 days post-injury as well, we identify, even in the young or the animals that were treated, a difference in the alpha diversity, and then as well in the genera and then the phylum. So make definitely an important impact on the microbiome. When we look at the different genera of the species, we identified only the animals, the Furnicuts and then the Bacteria, they were different with the animals that were treated with the PAM probiotics, but used only after injury. That didn't make an impact without the injury. And then several, then definitely was a dysbiosis that the PAM probiotics they was restoring. For example, the Alice tips or another kind of big family of all these. We want to look as well the metabolites that we feel that was important. So it's not only the bacteria that we identify with the cystic gas, but as well which metabolites they can produce. We didn't find anything relevant, any difference at 3 days after injury, but yes, 35 days post-injury. So that's meaning in a chronic state, the difference between the groups that were treated with PAM probiotics or not, they were more relevant, for example, in the acylbutyrate or the valate or all these compounds that are neuroprotective. So they were identified at high levels in after PAM probiotic. But as you see here, we always do males and females studies. So the males, they respond differently to the inflammation than the females. And curiously, only the probiotics works in males. So there are sex-dependent mechanism that is working here, and we need to try to figure out which one is. But there are only neuroprotective and high levels in males and not in females. So that is something that we can use for future studies, that the administration of the PAM probiotics to make that have some impact in neuroinflammation and recovery need to be chronic. So need to be administered for a long period of time. We want to look at the brain, if the impacts of the PAM probiotics, they reduce the lesion, and FED did it. And then we found here that you see here, this is I don't know if you are familiar with the brain that much, but when we have a lesion in the brain, we measure the area or the volume, and we found that the PAM probiotics reduce the lesion. Also, they reduce the cell death and the apoptosis. And then we found a reduction in the inflammation. That is the key point. We thought that we hypothesized that the mechanism that they regulate the lesion volume and everything is through the inflammatory response. So the PAM probiotics in these metabolites, they are reducing the peripheral systemic inflammation, and then that interferes with the brain, and we found the reduction of the inflammatory cells. And then, as well, we did the motor test to look at the motor improvement. In the animals, they were treated with the PAM probiotics, even males and females, in this case, both, was improved in the motor recovery. So they can walk better in everything using this device that is called the rotor. So that is the important thing. At functional level, the probiotics, not only in the inflammatory response, but functional level, they induce an improvement and the recovery after trauma. So basically, to summarize this first part, we thought that the TBI induced, of course, the change in the gut microbiome. This was the work that we published a few years ago. The lactobacillus strains definitely decreased the response of the effects of TBI. reduce antidiabetes inflammation. And then the probiotics and the lactobacillus family could improve the TB outcomes in a sex-dependent manner. That means only they were working this conjugation of the lactobacillus in males and not in females. So we want to explore a little bit more what we can do with the animals and the way that we can manipulate the gut dysbiosis. And for that, the best tool is use the germ-free mice. So animals that they board without, in isolators, without microbiome. So that is the best approach to see what happens if we induce inflammation in these animals and the way that they will recover. We didn't expect anything. Nobody else did this work before, so we want to run a pilot study with a few animals, germ-free mice. We did that in Baylor and the core facility there. And we induce TBI on the animals. And curiously, the animals, they don't have microbiome, they have more injury. So apparently, the microbiome, some way, is neuroprotective. So the microbiota and the animals, they don't have the microbiota, they have more inflammation, more lesion, and then they are definitely worse than the animals with the microbiome, with the regular animals, white type regular animals. So that is something that we didn't expect. We expect that the animals, they don't get microbiome, maybe could improve. But another approach that we want to do is try to generate these germ-free animals on the white eye animals in the lab, depleting the microbiome. So we use a cocktail on antibiotics in this case, and we know that the antibiotics is, in the clinical setting is something, of course, that the doctors use, and we don't know exactly the impact of the antibiotics in the gut and who this compound can help to the recovery or not. So that is the reason that we want as well run this second study and then provide some answer why the microbiota depletion benefits the brain recovery following brain injury. How much time I have? Like 2 minutes. Okay. So this is another approach that we did. So we did several. We induced inflammation in the animals in the beginning. We administered a cocktail on the antibiotics that they were quite aggressive. That means we're going to deplete all the microbiome. We're going to kill all these bacteria, and let's see what happens after in the recovery. And curiously, the first figure is showing that definitely the microbiome, the antibiotics deplete the microbiome 1 day post-injury, but 5 days after it starts to recover it, so the gut bacteria appears again. And we found that the TBI-induced change in the gut microbiota using the alpha diversity following TBI. But in the vehicle, we saw the impact of the TBI that makes a difference in the gut and the diversity, but not with the antibiotics. It seems that the antibiotics, they have kind of protective impact. And this is more complicated here, but for example, we found that after antibiotics, we saw a switch between the animals treated with antibiotics or not. Of course, we expect that, but 5 days after injury, we found that the group of animals with you can see here, treated with the antibiotics, they were not significant difference in the alpha diversity. It seems that they they recovered the gut microbiome diversity at this point, and we validate that with different analysis. So that means this is the point that we thought that maybe the antibiotics depleting the microbiome using the system has a neuroprotective role. And we identify that in the lesion volume, for example, the animals with antibiotics, they have less lesion, they have less cell death in different areas of the brain, in the cortex and thalamus, and etc. We identify as well less microglia activation, less inflammation. Using the antibiotics is neuroprotective in specific areas in the brain, and the animals, they recover faster. So that is a little bit contradictory to the previous results with the germ-free mice. So as conclusion, we can say that, yeah, acutely, short-term, the germ-free mice have a large lesion, and then, with antibiotic treatments, reduce the microglia inflammation. So maybe it's beneficial in the clinical setting for the recovery of the TBI patients, and as well the change on the microbiome. So that is kind of a summary that everything that we're doing. And finally, to conclude, we say that the change on the gut microbiome, we can use it as the biomarker for concussions or for TBI, because there are not good biomarkers, even there are no treatments. But as well, it's a good target to generate new therapeutic approach or combination of pharmacology with approach modulating the gut microbiome for future studies. And as always, need to be required like personalized medicine, because they depends on the age and depends on the sex, etc. So it can be beneficial or not. And that's it. I would like to thanks to my lab that we are here in the Research Institute, the Histometry Research Institute, and all the funding and collaborators. Thank you. Yeah, I don't know. You have some questions.
Any questions? I have a couple. Okay, so the one question I have is the previous talk focused on disorders in the brain, and you focus on microbiome changes linked to disorders in the brain. Would it be interesting to combine some of those results with structural variations going on in the brain with maybe processes observed in the gut? Or have you looked at any of this in terms of just a broader picture of what's going on for some of the disease processes?
Yeah, that will be very interesting, and we are trying to explore specific areas in the brain, the way that we can targeting and modulating the impact of that. In fact, we are using like nanoparticles encapsulating neuropeptides that are produced in the gut in specific areas in the brain to try to determine the function depending on the brain areas, and probably, but that requires more. I think that we still are in the initial stage, all the research, and future studies should be like that.
Thanks. And maybe something, Fritz, we can discuss in the panel discussion there. Any other questions, comments? Okay, over here, sorry. Question over here.
Oh, okay.
Hi, great presentation.
Thank you.
I was wondering what are the main differences between the recoveries between the probiotic and the antibiotic, and if you thought about combining those two therapies?
Yeah, in fact, we use that is very good question, because it took me like 2 or 3 years to reply. We use different strategies, and we don't know if it's convenient or not, do the microbiome depletion using antibiotics before a treatment, as can be like probiotics or fecal matter transplants, etc. We didn't did the antibiotic depletion before the probiotics, but we did it with the fecal matter transplants. When we use the antibiotics, the fecal matter transplant, like they lost the impact or the therapeutic impact. When we don't use the antibiotics, the fecal matter transplant works, and they work very well. So I think that this, is more, it's a combination. We need to figure out if it's convenient or not, deplete the microbiome before or not. And I don't think that the impact of the antibiotics is beneficial a long term. So that but we don't know exactly before the probiotics or not will be a good strategy. I don't think so. I think that there are different mechanisms that are working independently, not the same one. Yeah.
Thank you.
Yeah.
I don't know if I missed it in your figure, but how were the probiotics administered?
Oh yeah, I didn't say that, but it was in the oral.
Oral?
And the drinking water. So we prepare the probiotics daily, and we administer it for 3 months in the drinking water. So, and then we did the qPCR to verify that they were drinking.
Have you looked at how sustainable that is long term? Because I feel like I've read a paper a while ago that saw that oral administration of probiotics isn't long term. Like it doesn't change the microbiome permanently.
Yeah, in our case, we did oral gavage. That is oral gavage directly during 3 days, but that generates a lot of anxiety to the animals. So even we did with the vehicle, and then the drinking water, they are drinking the water like regularly. So we found major impact and 13-5 days, so chronically with the drinking water and not with the, but there are different ways to administer the probiotics in the food as well, etc. I think that if it's less invasive, you are not generating anxiety to the animals, and maybe the impact of the treatment is better. That's the reason that we did it. Yeah, thank you.
Thanks.